Well, hello. This is Lecture 1, Introduction to Microbiology and Aspects of Biochemistry, which cover in the textbook chapters 1 and 2. And so we're going to get started with this right now. What is microbiology? It's the study of microorganisms. We might commonly call them microbes, but the term really encompasses bacteria, archaea, fungi, protists, helminths, and viruses and prions, which are considered either non-living or non-cellular. When I talk about something prokaryotic, it lacks a nucleus and membrane-bound organelles. Example, bacteria. When I talk about something eukaryotic, it has a nucleus and membrane-bound organelles. Plant cell, animal cell, fungi cell. Keep in mind that there's an importance to the nucleus because it stores the chromosome, the genetic blueprint for the organism. Now, you notice here they give you a couple of classifications and some information. This is really important because here's the problem. When do you say that something's not cellular? And if it's not cellular, is it alive? And if it's not alive, how is it able to keep replicating itself? Well, first off, we have viruses. They consist of either DNA or RNA for their genetic instructions. They're surrounded by a protein coat. They're considered obligate, meaning absolutely got to happen there. Intracellular agents that use the machinery and nutrients of the host cell to replicate. Short explanation is basically this. Viruses have to have a cell to take over and use the cellular machinery to replicate more viruses. And in certain cases, the host cell dies. In other cases, it just kind of limps along, continuing to bubble off certain viruses. Now, viroid's different. This is really weird. Consists only of RNA. Basically, you've got a loop of RNA. It has no protein coat. It is an obligate intracellular agent that uses the machinery and nutrients of host cells to replicate. Most viroids have been found only in plants. But who knows? Maybe someday we find some in humans. Who knows? Prions are different. They consist only of protein. They have no DNA, no RNA. They are considered misfolded versions of normal cellular proteins that cause the normal versions of these proteins to misfold. That is how you have replication of prions in the cells. Now, when you look at different cellular organisms and non-cellular organisms, if you want to call it an organism, you have a classification that goes very similar. You can have a single-celled organism or a multicellular organism, but the question really is it prokaryotic or eukaryotic? Prokaryotic, and we'll get into that in the next lecture, basically is an organism that lacks that nucleus that contains the chromosome for the organism, and also lacks membrane-bound organelles. So you have bacteria, you have archaea. Now, archaea is a different grouping, and they live in extreme environments, high salinity, low acidity, very high temperatures. Some can live in the presence of all sorts of toxic material or at the bottom of oceans. But whatever you do, archaea have very unique living conditions, and their structure of the cells allows them to survive and thrive in those unique conditions. Protists are the first example you might think of for single-cell eukaryotic organisms, but some protists are actually multicellular. Now, by the way, we've got into this pathogenic, non-pathogenic, etc. Okay, bacteria can be both pathogenic and non-pathogenic. Protists, pathogenic, non-pathogenic. An example of that for unicellular would be for a protist, they're also called protozoans, is an amoeba. A multicellular example would be algae. Fungi are eukaryotic, they're unicellular and multicellular. They're pathogenic and non-pathogenic. There are some that are absolutely pathogenic organisms that cause athlete's feet. They would cause disruptions inside of the lungs and growing colonies there. And then there are those harmless ones but they taste great. And those are mushrooms. You might be a mushroom connoisseur. Mushroom, though, would be a multicellular example of a fungi. Helminths are multicellular, and many that we know of are parasitic. So they can be parasitic roundworms, flatworms, liver flukes, etc. We'll get more into that later on. Viruses, as we said, by definition, no cells considered non-living, but they can infect animal, plant, bacteria cells, and they can have a DNA or RNA genome. Prions were the most amazing and difficult to discover, in part because everybody thought, well, you got to have cells. If you don't have cells, fine. But no genome at all, no genetic information. Oh, no, can't be. Till Stanley Prusher proved them wrong. So it took his work in the 1980s. They can be, prions can be transmitted by transplant, by ingestion. Prions have been found to be transmitted between humans, usually by exchanges of tissue or even transplanted tissue. It was very difficult for some researchers because they would call this slow viruses, but there was no evidence of genetic material. It's when we really understood the refolding of these organisms that we began to really understand what was going on. They have found that there are some prion diseases that are inherited, and that leads to other interesting research areas. So here's the microbial world. The acellular infectious agents, prions, viruses, viroids, the organisms, those that are prokaryotic, bacteria and archaea, those that are eukaryotic, which include algae, protozoa, fungi, and helminths. And we'll just move on from there. There is a nice page that gives you some perspective of the onset and the development of the science of microbiology, who developed what, when, and as you can see, as we began to understand things better, we began to connect certain organisms with certain pathologies, certain diseases. This was partly because of the germ theory, which was developed in part by Robert Koch. We also had the concept of the cell theory by Schleiden, Schwann, and Virchow that contributed work. And then seeing this in a medical arena, you had the work by, of course, Ignaz Semmelweis, who implemented hand washing to prevent childbed fever. Florence Nightingale that established the formal aseptic practices in nursing. Louis Pasteur presented his findings against spontaneous generation and showed that organisms do reproduce other organisms, sort of the biogenic theory. And Joseph Lister published his aseptic surgical techniques. Now, you don't have to memorize all the little details here, but I would like you to get somewhat familiarized with some of their work. This also brings up a very important area, and this is the endosymbiotic theory. You remember a while back I just said prokaryotic, eukaryotic, okay? How did you get cells that had organelles in them. How did they form this nucleus, this membrane-bound structure that contained all the DNA? This is where we get into a theory, in part developed by Deduve I remember seeing one of his papers in '69, and it's the evolution of eukaryotes. And he basically said that this was due to the symbiosis of prokaryotes and the host and the guest. The guest was the one that was kind of engulfed by the host. They exist together. They live together, symbiosis. Now, what is the evidence for this? Well, if you look at the organelle, the mitochondria, it has its own separate DNA. It has ribosomes inside of this organelle that are more akin to bacteria than they are to eukaryotes. It also has some of its own specific components of RNA. So it seems to be that it was some sort of ancestral, formerly an ancestral free-living organism that got swallowed up. But think about the arrangement here. Host gives shelter and nutrients to guest. Guest is the mitochondria. Mitochondria takes the nutrients and turns out lots more ATP per molecule of glucose. So mitochondria benefits from being basically shelter, nutrients. It gives out tremendous amount of ATP. Host gets ATP and provides shelter and food for the symbiotic guest. Now, we do have to go over terms. You have to be familiarized with all sorts of terms. When we talk about pathogens, these are disease-causing organisms. An opportunistic pathogen is an organism that is pathogenic, but this is due to the opportunity of a weakened immune system of the host. As the previous chart showed, the golden age of microbiology started around 1850 and went to 1920. And some of the milestones include the cell observation. That was the work done by Robert Hooke, who was able to say, look, these are cells. And they were actually more the cell walls of a slice of cork. Then we had Antoine van Leeuwenhoek, who his work with the real first microscope was able to observe bacteria. And here is his simple microscope. The specimen holder was the tip of that needle. The focus screw would move the needle back and forth, or the plate back and forth, with the lens until you could see something. Light would come in through this area here, through the lens, and be observed. And then they put a handle on it so that it could be held, etc. And here's another example of it. The handle, the focus screw, the specimen holder, and of course the lens. Now these are some of the basic drawings that were given by van Leeuwenhoek when he was able to say, look, you know, these things are moving around in here and I didn't even know they were there. And he notes this almost spirillum type of organism and these other small round or oval shaped structures. You can see these two here almost look like cocci of a bacteria. This was a whole new world. Now, a couple of other important steps on this road to the golden age: spontaneous generation and Francesco Redi. Redi in 1668 did an experiment, and this was the first controlled experiment where he took containers and they had meat in it that was rotting. Without a cover on the top of the container, flies came in, laid eggs, and eventually you start seeing all these little maggots. He took another exact same container, same time, put in meat, but put cheesecloth in it to prevent the flies from laying their eggs. The meat still rotted, but you had no formation of maggots. Now, to help you to understand this, now it sounds ridiculous, but this is partly what they had. Spontaneous generation would say anything that was non-living could have the potential to generate living matter. So you'd look in the mud of a river and you'd see, there are eels there. It must be that the eels were generated out of the mud. You would look at, rotting meat and say it would generate maggots. It was a difficult theory to get rid of. The final defeat of this theory was by work done by Louis Pasteur in 1864, where he took broth and he had these very long tubes from basically a flask that contained hay broth, and if you boiled it and you had the container open, and what I mean by open is no long neck, you would have bacteria that would come in and colonize and begin to grow in the water. The liquid medium, the broth, would go dark. But with the long necks, it prevented organisms from going up through the tube and into the sterile medium. And it's said to this day at the Pasteur Institute in France that they still have some of those very same flasks. And after 100 years, they're still sterile. Now, this is where he led to what was called the biogenesis theory. Life comes from life. This is summarized by the phrase omni vividium ex vivio. Latin for all life is from life. A related statement that omnia cellula e cellula, all cells are from cells. In other words, what he's basically saying is living matter gives genesis to living matter, not non-living can spontaneously form living matter. Okay? This conclusion is one of the central statements of cell theory. Here's the Francesca Redi experiment. Disproves the theory of spontaneous generation. Here we have the flies. Here we have the maggots. Here we have an open jar. Here we have a gauze-covered jar. No maggots appeared on the gauze-covered jar. Here we have a sealed jar, completely sealed. No maggots appear in the sealed jar. But because of the onset of the development of the observation and the discovery of microorganisms, bacteria, some people said, well, maybe what it is they're just too small to see. So we go to Pasteur's work, where he takes broth, sterilizes it. The trapped air escapes from the open end of the flask. Notice that it's a loop. So bacteria and dust from the air settle in the bend of the loop. But years could go by and the broth stays sterile indefinitely. Now, what he did to prove this also was the flask could be tilted, that the sterile broth comes in contact with bacteria and the dust from the air, brings it back in, and then with a short number of hours or days, the bacteria multiply in the broth. Pasteur's work also developed pasteurization. Now, it may not mean much, but I want you to think about this. He found that if you heated wine to 50, 60 Celsius degrees, it killed the yeast and the bacteria that usually cause wine turning bitter while in storage. We do this all the time now. Milk, food, fruit juice. Pasteurization, for example, with milk. Sometimes what it is that they run it through a very, very hot tube for a very small time, something like 165 degrees Fahrenheit, and it's run through the tube. The length of the tube and the speed in which it's passing is basically exposed for 15 seconds. Boom. It's safe to drink. It's safe to store. It will not go bad. The germ theory of disease developed this concept where microbes cause infectious disease. This was competing against the other concept, which was the bad air, which causes disease. This is where we get the word malaria from. Mal being bad, aria, air, vapors. In other words, it was not a mosquito bite and a parasite that made you get malaria. It was you were around the swamps and they had bad air and noxious air or whatever, and that was why you got malaria. Well, Robert Koch, the German physician, developed staining and culturing techniques. He's best known for that, but he's also known for the germ theory connecting anthrax to Bacillus anthracis. He could isolate the bacteria from animals and purified the bacteria to infect other mice. You see here, there's a 4-step process. Basically, what he says is that if this organism causes the disease, 1, you have to find the organism in diseased individuals or animals, whatever you want to say. You remove that organism and you culture it. You isolate it from the diseased host and grow it in pure culture. Then you take healthy animals and you inject in that culture of the suspected host, the suspected pathogen, I should say. The formerly healthy animals become sick with the inoculated organism, and when you eventually do an autopsy, the same pathogenic organism must be there. And you could even remove it and isolate it and culture it again. What he's basically doing is showing a process where you can connect organism with disease. Now, there are some technical challenges. Some organisms cannot be easily cultured outside of an organism. Treponema pallidum, sorry, I do stumble once in a while. This is the organism that causes syphilis, and it cannot be cultured in glass, that is in vitro, in other words, grown in petri dishes and things like that. It can only be grown in vivo, in other words, in animals. One of the typical situations is that they will infect rabbits to do research on syphilis. Now we have another situation, historically much closer in our time, HIV. Human immunodeficiency virus was not easy to culture and could be only cultured in monkeys. You couldn't do humans because, hey, you're giving them HIV, and even in the early days, they knew that was a death sentence. Prions, a lot of the prions that have been, that some prion studies have been done, you infect hamsters and you can isolate the strains and you can replicate this. So there's a way to deal with cautious postulates, but you can't always just cultivate them in glass outside of an organism. Here is Treponema pallidum, and you can see it's a spiral shape organism, bacteria. Now, next thing. We're going to be dealing with a little bit of this later with hand hygiene and aseptic technique. Of course, in the age of COVID-19, that's become a big issue. When I talk about aseptic technique, these are procedures and methods to prevent infectious agents from being introduced to a patient. Also, the term is used in the procedures to develop a pure culture, prevent cultures from being contaminated by other organisms. When I say something is sterile, it's absent of all microbes. Now, in a hospital, aseptic technique is used to prevent hospital-acquired infections, HAIs. They used to be referred to as nosocomial infections. Now they use HAI and basically refers to infections acquired in a healthcare setting. So it's not just a hospital, could be a nursing home, could be a hospice care or anything else like that. Some of the keystones of work done by scientists to deal with and really help build the foundation for aseptic technique, it includes Ignaz Semmelweis in the 1840s, decreased childbirth fever, which is really a staph infection from unwashed hands. You have to understand that these were physicians who, in 1 hour, they would be doing an autopsy, and then the next hour they would be assisting in the delivery of the birth of a baby. No, they didn't wash their hands. And so the women, the mothers who had just delivered the babies would die, sadly, of these infections. Now, Lister used carbolic acid, which we now call phenol, on wounds. It's kind of tough on wounds, but when you got nothing else. Remember, this is the 1860s. He also used this as a disinfecting agent. So in other words, he found that sterilizing instruments and sanitizing wounds would help prevent infection. Now, Florence Nightingale had established aseptic techniques in nursing practices, along with other patient care practices. Yes, that's the founder of modern nursing. Now, we have to also step over aside here and review the basics of scientific methods used to illuminate microbiology as a science. Now look, we got no. Leaching and bloodletting and all these other things, that was, 13th, 14th, 15th century, that type of stuff. What scientific methods or the methodology is that it starts with a question that can be investigated. You see a particular event. You set up a hypothesis. This is a testable explanation to what you observed. A hypothesis really is a prediction or proposed answer based on prior experiences or observations, but this is the key point. It has to be tested. In other words, if I came to you and said, hey, the reason why the flowers open up every morning is because you have little pixies that go up and tap it with their magic wands. Yes, everybody would think I was nuts. But here's the point. Why do you think it's nuts? Because we've never seen, measured, observed, captured pixies. You can't go down to Walmart and get 5 pixies for $1 or 3 pixies for $2, whatever. The point is you have no way of measuring, etc. So you have to have something that can be tested, observed, measured, and this data, these observations that you collect after in this testing, will help you to formulate a conclusion. Now, basically, the scientific methodology will state whether the hypothesis is contradicted or rejected. We never, ever say proved. You would say that either the hypothesis, the explanation of the observation, is rejected or that it's supported. Here's part of the point. Lots of people say, I have a hypothesis, but they don't know when and how to basically test it. And if the test fails, they're not willing to accept it's contraindicated or it's rejected or it's supported. They immediately go and just say it's proved. You can't prove something. Why? Because later on, you have to keep an open mind and have the potential to understand that maybe a better measuring system, a better test, may eventually reject the hypothesis. The key point is keep an open mind, because you may find other means of observation, new measuring tools that can refute the hypothesis at some later time, but at the same time lead you to further knowledge. Now, we have to also limit inference observations. That's this jumping to conclusions. Okay? All data is always collected by our senses and by our instrumentation. Let's face it, we have instrumentation that goes beyond our senses. You can see, you have devices that can image in X-ray, ultraviolet, infrared. I can't see ultraviolet or infrared, and neither can many people I know of. But we have that capability to measure certain things in areas that go beyond our immediate senses. Therefore, we always have to be aware that we're always limited to our observation tools. Just one little point to help you to understand it better. If you go back 100 years, there were some devices around that might have been able to tell you if you took a drop or two of dye and put it into a swimming pool, you'd be able to detect a couple of those dye molecules. Today, we have the sensitivity of techniques that could determine if you dropped a couple of drops of dye in a vast ocean almost, under the right conditions. Yeah. Now, let's also get to understand what these terms, law and theory, are. A law is a precise statement or mathematical formula that predicts a specific occurrence based on carefully limited and defined terms. Basically, it's looking back at prior data and says, this is what all the prior data collected supports, predicts that this event will occur or this outcome will occur. A theory is a hypothesis that has been proven, that's supported, through many studies with consistent supporting conclusions. Theories may unite different hypotheses and laws, but keep in mind, anything that's a hypothesis can be overruled or overturned, with some other experiment down the road. When we start looking at bacteria, it's not just a matter of saying bacteria. What are they? Well, they're single cell. Yeah. Well, what do they look like? How big are they? Are they single cell, meaning they don't hang around with other cells, or what? Morphology refers to shape. Physiology refers to, let's say, biochemical function and things like that. So when we talk about morphology, we're talking about physical structure, shape, size, arrangement. There are limits for what we can understand for bacteria and other organisms. We have to do several approaches. Sometimes if you sit there and you say, okay, I've got these blue specks. Okay, that's gram-positive. But what are they? Well, what do you mean? Well, are they this type of bacteria, that type of bacteria, what? We'll get more into the processes of tests and other types of techniques later. What helps is if we understand that with unicellular, that is one cell, organisms, they can be bacteria, archaea, or simply eukaryotes. They just basically are all on their own. They have a nucleus. They, swim around, float around, do whatever. Multicellular, they're usually many cells. And a lot of times these cells are going to specialize in structure and in function, which allow the eukaryotes a lot more sophistication, not only in their morphology, but in their physiology, their biochemistry, their genetic makeup, and in their fitness, their survival in the environment. We'll see some of that down the road. When I talk about taxonomy, this is the study of how organisms can be grouped by shared features, and the process encompasses identifying, naming, and classifying organisms. Today, the techniques organize life forms by shared characteristics of physical and biochemical features, by their ecology, and by gene sequences. One of the best ways that we've known is ribosomal RNA sequence. Now, if you remember what ribosomal RNA is, the ribosome is an organelle that helps synthesize proteins. A ribosome is composed of a smaller subunit and a larger subunit. And usually, the ribosomal RNA sequence can handle a lot of changes, because remember that the ribosomal RNA is really sort of like this framework structure inside the ribosome, and all these proteins glom on or attach to it to form this kind of, globular structure. But if you have a mutation or two in the wrong way, ribosome is not functional anymore. Today, we have identified many different life forms and their similarities and relatedness, etc., via ribosomal RNA sequencing, and been able to separate out some of the differences based on ribosomal RNA sequence. Now, where do we get all these names for organisms? Well, Carl Linnaeus devised the binomial nomenclature system. It's really two names. One is for the genus, one is for the species. So when we hear E. coli, the full name is Escherichia coli. And by the way, the first letter of Escherichia, or if you abbreviate it, E, is capitalized. Coli is all small case, but I'm making this easier to read. Escherichia coli was first isolated by Theodore Escherich. It's a bacteria that's located in the colon. There are some strains that are totally harmless. We use them as sort of the microbiology lab rat in lots of experiments. There are some other ones that are very, very dangerous because they can cause serious illness or death. Now, here's a couple of examples here for scientific names. Haemophilus influenzae, Saccharomyces cerevisiae, Shigella dysenteriae, and Staphylococcus aureus. I would give you some time to review that. Not necessarily you got to memorize everything, but just basically you can see where some of the names derived from certain properties, certain physiological effects, certain diseases they might cause in those that were the discoverer of the organism, etc. Okay? Or certain shapes or other characteristics. Now, I'm showing you here basically the big three divisions here. You have bacteria, you have archaea. Bacteria and archaea are somewhat alike. They're both prokaryotic, single-celled. But their ribosomal RNAs are different, and where they live are different, and their cell wall compositions are different. Then we get to eukarya, and they're very different from bacteria and archaea. And the cell wall composition is different, the ribosomal RNA nucleotide sequence is different, etc. Now, the taxonomic hierarchy, also developed by Linnaeus, in biological classification, this is sort of a taxonomic rank. It's sort of the relative level of a group of organisms. And a group of organisms, in this case, would be referred to as a taxon. Each level defines select characteristics and relatedness. So when we talk about domain, as you saw in the last slide, that is the broadest sort of classifications. And that's basically bacteria, archaea, and eukarya. When you used to have the 5 kingdoms, this was overturned by Wollasey's work with ribosomal RNA. But beforehand, 5 major kingdoms were sort of the division of life. That was plants, animals, bacteria, fungi, and protists. Now, we have these lower levels of classification, and it's sort of what's compared with what at the phylum level, what is similar to what at the class level, what is similar or different in the order, in the family, in the genus, in the species. Always remember at the end, it's genus and species for the particular name, for the particular thing. Now, one other thing, the strain. This is sort of after species. It's a term used to recognize genetic variants to the same species. Different strains of different species have a key genetic trait that warrants a special designation. So they'll either be numbers or letters or both. For example, E. coli K12, relatively harmless. They use it in a lot of different experiments. Like I said, it's your typical lab rat experimental bacteria. But E. coli O157 colon H7, that is the one that can kill you if you imbibe, if you take it in, such as in contaminated hamburger meat. You may have heard of cases where in the 90s, 2000s, individuals got very sick and in some cases died from what seemed to be just minor food poisoning, but what happened was undercooked hamburger or contaminated with fecal matter that contain this particular strain. And what this does is it will cause gastrointestinal damage and then eventually lead to damage that will then take on in the blood and in the kidneys, etc., and eventually kills individuals. Now, here you can see as you move from domain to species. Domain is least specific. As you go down each one of these steps, you get more specific. You notice here some of the differences right off the bat with life. unicellular, multicellular eukaryotes, unicellular prokaryotes. Notice that we get down to the genus, and for this particular organism, Clostridium tetani, the causative agent of tetanus, we have Clostridium as the genus name and as the species tetani. Yes, there are other species of Clostridium, and they do other things, so there's where you have the differences showing up in the species name. Now, host microbe interactions. You might be wondering to yourself, huh? What? We're going to get into this. How do organisms interact with the host? You, me, dogs, cattle, okay? Well, microbes, some of them are totally harmless, others are pathogens. A pathogen is an agent that causes disease. A symbiotic relationship, two or more organisms that live closely connected. Now, there's different types of symbiotic relationships. One is parasitism. The host is hurt, the other organism benefits. Mutualism. Both organisms benefit. Commensalism. There is no perceived benefit or harm to either organism. They just live together. An example of the parasite would be protozoans and helminths with complex life cycles. They live either on the exterior or the interior of the host. Then we have to get up with another concept called biofilms. These are sticky communities developed on a surface by select bacteria that secrete a protective matrix in which the bacteria grow, multiply, and are protected from various bactericidal substances, so antibiotics, disinfectants, etc. And because they have kind of clustered together and encapsulated themselves into some of this matrix, they are kept away from many of the environmental factors that would be used to destroy them. Here you see an example. Biofilm, an example of that would be dental plaque. Or biofilms develop on catheters, contact lenses, water filters, cutting boards. You have a few of them attached to a target surface, then they achieve a certain number and they start releasing the sticky matrix. And then you have a buildup of this biofilm. Some will split off or free floating. They form what is referred to as planktonic, and they spread off to form other biofilms at other places. And you can see this here on a dental floss with biofilm scraped from the teeth. Now, other host microbe interactions. You have normal microbiota. Now, what do I mean by that? We also refer to as normal flora. These are common microbes, bacteria, archaea, simple eukaryotes. They're found in various parts of the body, but they don't trigger disease. Some even enhance our immune system. They may produce vitamins, or they may help us digest food. The establishment of human microbiota occurs at birth, usually through skin contact, breastfeeding, the environment after birth, or prior to birth in utero via the placenta. Now, during disruption of normal flora, a specific part of the body, such as the intestine, may become host to other microbes. Here's part of the point. The normal flora, the normal microbiota, may actually create the conditions to make it hostile for certain pathogenic agents to thrive in that environment, whether that's in your colon or your sinuses or the vagina or whatever. But when an individual starts obliterating or knocking down the normal microbiota, the protective characteristics are lost. And there you're going to start having other pathogenic organisms become proliferating and taking over and leading to disease. Here is an example of the different types of normal microbiota and the species around in the intestines, the urogenital tract, the skin, the mouth, pharynx, and upper respiratory system, and even in the stomach. Don't worry, you don't have to memorize all of these now, but you will, as the course continues, get familiarized with some of them. But it is important to realize that we are not completely sterilized and clean and absolutely free of all microbiological organisms. We just happen to have some of them that are on us that don't cause any disease or any problem. Now, host microbe interactions include the transient microbiology, contact with formites. Now, formites are things that we come in contact with. Handrails, doorknobs, door panels, or a handshake can exchange microbes. That's why everyone's getting very concerned about COVID-19, the common exchange from one hand to another. And so we're told not to give handshakes. But good hygiene can remove these organisms. Some microbes play a key role in the reduction of pollution. For example, there is the concept of bioremediation. That's using specific microbes to remove or detoxify toxic waste or pollutants. When they've had situations with oil spills, they have spread out either the organisms themselves or the nitrogen and phosphorus necessary to cause these organisms to become very prominent. They break down the petroleum products and then basically just disappear. Now, we're going to move into the other area, which is involving biologically important macromolecules. This is kind of a brief study. You've dealt with this before in other courses that had biochemistry as one of the early chapters. This is just a glossing over to just go over some important areas. The four main classes of biological molecules, biomolecules, are carbohydrates, nucleic acids, lipids, and proteins. Biological macromolecules are built by synthesis reactions and broken down by decomposition reactions. In other words, you synthesize, you build up the simpler molecules to make much more complex molecules. You have decomposition reactions breaking down the polymers back to their simple building blocks, monomers. Okay. The concept of polymerization, this is the process of covalently bonding monomers to a macromolecule. Example, glucose being made into glycogen. Glucose being made into cellulose. What is cellulose? Cellulose is just big long strings of glucose molecules attached to each other. Let's start off with carbohydrates first. Carbohydrates, actually they were once called more hydration of carbon, are basically have a carbon to hydrogen to oxygen ratio of 1 to 2 to 1. All have one or more sugar group. Monosaccharides, these are basically single sugar groups. Glucose is one, galactose is another, okay, fructose is another. Now, if you take, for example, fructose and glucose and have a covalent bond between them, you have a disaccharide. A disaccharide is basically a simple molecule consisting of two glucoses, two sugars. You can have glucose and you end up with maltose. You can have galactose and glucose bonded together and you have lactose. A polysaccharide consists of more than two sugars held together. Glycogen, amylose, otherwise known as starch, cellulose, etc. The chemical bond is called a glycosidic bond. It is a covalent bond that forms with the loss of water. The functions of these type of carbohydrates, well, cellulose is a major structure in plants as well as in algae or algal cell walls. Chitin is another type of cell wall component that you see with fungi. Peptidoglycans, and these are basically macromolecules of protein and carbohydrates combined, and they form cell walls of bacteria. Also, some carbohydrates are involved with cell signaling. Bacteria will use the carbohydrates to communicate within and between cells. And you can see this display of the four main classes of biomolecules. I encourage you to take some time and do a little bit of review of this. Next we're going to deal with is lipids. These include fats, oils, steroids, and waxes. They have a carbon-hydrogen-oxygen ratio, but usually what you're going to find is that it's high carbon, high hydrogen, but a very low amount of oxygen. As a result, many of the lipids are hydrophobic, okay? And that means that they will not associate very well with water. A saturated fatty acid chain, it has no double bonds in the fatty acid chain. An unsaturated fatty acid has one or more double bonds, carbon to carbon, in the chain. They remain liquids at higher temperatures, which is interesting. Monounsaturated has one double bond, polyunsaturated has two or more double bonds. Triglycerides. This is a molecule that's formed where you have three, that's where you get the tri, fatty acid chains that are attached to a glycerol molecule. This attachment is using an ester bond. Each chain is attached generates one water molecule when you actually have the chemical reaction to attach the chain to the glycerol. When you have diglycerides, you have two fatty acid chains attached to the glycerol, and monoglycerides, one fatty acid chain. There's an interesting type of lipid called sphingolipids, which link fatty acids to sphingosine. You'll see that in nervous tissue, for example. Here's an example of a saturated fatty acid. If you'll notice the chain here, it's pretty much single covalent bonds all the way through. And as a result, in this triglyceride, all the fatty acid chains are all straight. As a result, they get easier to stack, and they have a much lower melting point than unsaturated. Saturated fats you see as butter, sold as solid at room temperature. Unsaturated, because of the presence of the double bond, causes a kinking in the chains. These kinks lead to the molecule not being able to easily stack up, and therefore its stability is going to be more in a liquid state than in a solid state. It would take a much, much lower temperature to take the unsaturated fatty acid and return it to a solid, okay? Whereas at room temperature, the saturated fatty acid is a solid. Therefore, when you look at unsaturated fatty acids, they're usually in a liquid form, okay? At room temperature, I should say. Here is an up-close example of fatty acids, triglyceride. Now, when we talk about lipids, we also talk about waxes, which are lipids derived from chemical bond between a long-chain alcohol and a fatty acid. Usually, these are semi-solid to solid at room temperature. Yet, polyunsaturated wax, such as jojoba oil, is a wax but a liquid at room temperature. Okay? So you can have examples of wax-like compounds, which we'll get more into in cell walls of the prokaryotes in the next chapter. And as a result of being liquid at room temperature, some of these waxes will play a role in actually cell walls or cell membranes of some of these prokaryotes. Now, steroids. These consist of 4 fused hydrocarbon ring structures. Common example, cholesterol. But cholesterol is really a modified steroid called a sterol. That's because it has an alcohol functional group added on one side of the molecule. Interestingly enough, adding an alcohol group makes the sterol ambipathic. Now, you have to understand what the term ambipathic means. Heads up, could be asked. You actually have a molecule that's partly polar and partly nonpolar. You see this all the time. What do you mean? In detergents. In detergents, you have, they are ambipathic molecules. They interact on one side, the polar side, with water. They interact with the nonpolar side, and that's usually the grease or the dirt or whatever, and it works out wonderfully. Okay. Lipid functions. They are a source of energy. They are a cell's structural component, and they mediate cell signal. Glycolipids, for example, this is where you have a carbohydrate attached to a lipid. These are essential for cell-to-cell signaling. Lipoproteins, where you have a protein attached to a lipid. That's another example. Glycolipids, when we get into this, you're going to get familiar with one, lipopolysaccharide. This is present in gram-negative cells. It is toxic to animal cells, serves as a protective barrier for bacteria, and helps bacteria adhere to surfaces. When we talk about waxes, they serve for protection. They're also an energy source. One interesting wax, mycolic acid, has been found in tuberculosis bacteria, and it acts as a protective barrier and enhances bacterial pathogenicity. Here you have an example of a wax. This is a long-chain alcohol attached to a fatty acid. Boom. Guess what? This is the formula for beeswax. Notice this one here, where you have the attachment. This is the long-chain alcohol. Okay. This is the fatty acid component, and this is the molecule for mycolic acid. Steroids, as I mentioned to you, you see the 4 rings here, and off of them are functional groups, such as a methyl group here, a hydroxyl group here, or a ketone group right here. In this case, you've got 1, 2, 3, 4, 5, 6, 7, 8. So you'd have an octane or octyl group attached off of this one. This is cholesterol. And as a result of having this OH group here, it's a sterol, not as much a steroid. But here's the point. The main 4 rings give this certain properties, as is included in the lipid family of compounds. Here you see a photo, and you can see some of the lines of the mycolic acid that make up this particular tuberculosis bacteria. Now, the next one we're going to deal with is nucleic acids, best known DNA and RNA. They're macromolecules. They serve as genetic material for cells and viruses. Deoxyribonucleic acid, DNA, is a double-stranded molecule. It has deoxyribose for sugar, and its bases on the inside would be adenine, thymine, cytosine, guanine. Keep that in mind, because ribonucleic acid is different in the sense that it is single-stranded, has ribose for its sugar, and its bases are adenine, uracil, cytosine, guanine. Nucleic acids are polymers of nucleotides. The nucleotides have to consist of 1, a 5-carbon sugar, so that could either be deoxyribose or ribose. 2, 1 to 3 phosphate groups are attached. And 3, a single nitrogenous base. That could be either adenine, guanine, cytosine, thymine, or uracil. Nucleic acids are held together by polymerization of the phosphodiesterase bonds. These are the covalent bonds between phosphate and the sugar. Such as here is the nucleotide, here's the phosphate, here's the sugar, here's that base that we have to be clear on. Now the sugars could be either ribose or deoxyribose. That would be there. And then we get into the nitrogenous bases. They all have nitrogen in them. And you either have basically adenine, guanine, or cytosine that will be present in either DNA or RNA. The difference is uracil will be present in RNA. Thymine is present only in DNA. And you can see how this assembly occurs, where you have a dehydration synthesis and the formation of the phosphodiesterase bond. So you continue with this backbone of sugar, phosphate, sugar, phosphate. And the bases are on the on one other side. Now, I was going to say on the inside, because when you have a double-stranded DNA structure, all the bases are going to be inside. And like a staircase, a spiral staircase. Also in nucleic acids, we have to consider adenosine triphosphate. That's the energy molecule for cells. When the phosphates are cleaved off, energy is obtained for cellular functions. What is the function of these nucleic acids? Well, DNA is the genetic blueprint for all cells. DNA and RNA can be the genetic blueprint for viruses. Ribozymes, interestingly enough, are strands of ribose of RNA that are specialized so they can catalyze particular chemical reactions. Now, some ribonucleotides are chemically modified and used in cell signaling pathways. Finally, we get into proteins. Now, proteins and their building block is amino acids. You have to start getting used to some of the terms here. Proteases are specialized enzymes that break peptide bonds by catalyzing hydrolysis reactions. Polypeptides are long chains of amino acids, anywhere from 55 or greater, the number of amino acids. Peptides are usually shorter chains of amino acids, less than 55 amino acids. And due to the diversity of amino acids and possibility of long chains, proteins can take on different levels of protein folding and thereby have a greater variety of three-dimensional structures, okay? The protein folding process, where proteins, higher order of protein structure, this depends on the folding orders as it relates to the function. The structure relates to function, vice versa. So we're going to see this in a second. There is another type that's important because here's the thing. What happens if proteins inside of a cell get worn out? Well, this is where we get into chaperones. Chaperones are smaller proteins that help larger proteins achieve their conformational folding to achieve tertiary or quaternary shape. And chaperones, it's interesting to note, when some cells get exposed to heat, they start to have the loss of some of their tertiary or quaternary shape. It's interesting because chaperones were called heat shock proteins until it was understood these two different groups were really the same thing. And chaperone numbers increase when there's been some influence of heat. And basically, think of it as a bunch of people just moving around, putting things back together inside of a very, very busy, let's say, room or conference center. They kind of clean up and get everything set up neat again. All right. What is some of the functions and what are some of the other important details we have to talk about when we are reviewing proteins? They're literally the cellular workhorses. They're building blocks are amino acids because there are over or about there are 20 different amino acids, but there 22 are genetically encoded. You can have all sorts of different properties, sizes, structures, etc. Now let me help you understand that. If you look at an amino acid has sort of a central carbon. It's got kind of on top of it a little hydrogen. It's got one side has got a carboxyl group, the other side's got an amino group, and then there's the R group. And that is the group that results in the diversity of different amino acids out there. Now, R group can be simply just a hydrogen. It can be a very long hydrocarbon chain. It can be a multi-chain structure. It can basically be a thiol group. It can be all sorts of neat different structures. Now, the diversity of 20 amino acids and then two other non-standard, selenocysteine and pyrrolysine. This is because they're the way they're encoded. Give you all sorts of diversity of structure and function. And by the way, the size of proteins can be, we talked about 55, we talked about smaller numbers, but they can be up to 10,000 amino acids, be incredibly large structures. Okay, so amino acid chemical bonds, That's covalent bonds. They're called peptide bonds. A water molecule is lost in the synthesis of this type of bond. It's called a dehydration synthesis. Now you see all these green areas here? This is the R groups. R is basically to designate that there is a variety here, and you have to go and seek out what is the particular type. Some provide polar properties to that particular amino acid. Some are nonpolar. Some are more acidic. Some are more basic. Some are non-standard, and that's where we get into the selenocysteine and the pyrrolysine. You go back to this basic structure for amino acids, as I said before, central carbon, carboxyl group on one side, amino group on the other. There's sort of this little hydrogen here, but it's the R group. And you have to understand that the R groups provide certain structure and physical properties. Is this protein going to be water soluble? Is it not going to be water soluble? Can it take a large amount of heat? Is it going to act as an enzyme? Is it going to act as an antibody? Is it going to be a cell receptor? All sorts of different things. Now, let's go through the structural levels here for a minute. Primary, this is basically like beads on a string. Amino acids, one attached to another, touched to another, touched to another. A very linear sequence of amino acids in a protein. They're held together by peptide bonds. They can be altered by mutations, changes in the DNA code instructions for a specific amino acid. But for the most part, since they're all held together by covalent bonds, they're very stable. Secondary structural level, that's where sections of the peptide chain is stabilized by hydrogen bonds between sections of the string. Now remember, all those R groups, well, the other thing that you got to keep in mind is that each amino acid has sort of each of those amino acids has an amino group and has a carboxyl group, and as a result, amino and carboxyl groups can interact and form hydrogen bonds. This leads to these localized sections that are going to have different shapes. For example, the alpha helix, it's kind of like a corkscrew, a spiral. Or the beta pleated sheet, it's like an accordion-like fold. It's just like folding over on top of it again and again and again. And these will be usually multiple sections on the single peptide string. Now that's secondary. Let's go to tertiary. This is where you now get larger sections of the peptide string folded, held together by various molecular interactions. This is where you get R groups playing a role, but also you will have other forces, van der Waals forces, ionic bonds, hydrogen bonds. Now, all of these type of bonds, just like with the secondary level, they're all not strong. They're not going to be covalent. And if you know what hydrogen bonds are, they're easy to separate by just adding a certain amount, a small amount of energy. Here's the primary structure. You can see amino acid, connected amino acid, and basically these two amino acids form the peptide bond right here between this carboxylic acid and this amino group, and it goes all the way down. Note where these R's are. Sorry, no joke intended. Secondary structure, they have these folds, the beta helix. What stabilizes them? Hydrogen bonds between one section and another section. Beta pleated sheets, it's almost as if you fold over. And so this will be connected to the next layer up on top of it. That's pretty cool. Yeah? Well, you want to see something cooler? When you get to tertiary, then you have all sorts of interactions going on, but then you've got one last one, quaternary. Now, quaternary means that you have to have two or more protein chains. Each of these chains would be called a subunit. And so what you have is a functional protein. When it has two or more proteins together as subunits, that means that you're going to have, again, some hydrogen bonding or something else to hold them all together, but they will only really function when all of their subunits are together. Sometimes you'll refer to them, if they have 2 subunits, we call it a dimer. 3 subunits, a trimer. 4 subunits, a tetramer. Stop right there. What's an example of a tetramer? You've dealt with it before. It's called hemoglobin. It has 2 alpha chains and 2 beta chains. And the only time that you really have functional hemoglobin is when you have all of the chains working together as a functional unit. The 2 subunits of alpha, the 2 subunits of beta, and that's where you're going to have the attachments of the oxygen molecules. Also, you can have quaternary structure present in cell surface receptors, enzymes, antibodies, etc. Remember, structure relates to function. Function relates to structure. Protein functions include structural scaffolds in cells, enzymes, cellular transporters, cell recognition, and antibodies. Don't forget the antibodies. Here's an example of the tertiary structure. Notice that you've got a disulfide bridge here, and what happens is you have that beta-pleated sheet effect. You got that corkscrew of that alpha helix, and it's waving over here, etc. Now, that's if you only have one. What if you had to have a functional protein that was composed of 2 or more? Here's what it would look like. Okay? Now, what is denaturation? It's the process of removing a 3D structural design of a protein with the subsequent loss of protein function. Remember I said structure relates to function, function relates to structure. Lose the structure, you lose the function. Lose the function, you lose the structure. Can you give me an example of that? Yeah. Remember, hydrogen bonds are weak. They can be broken down by moderate application of heat. Now, there are other ways to do it. Chemically, you can do it. You can add things like formaldehyde, urea. You can add acid. You can add bases. You can shift pH changes. You can add heavy metals. Why do you think they used to have in paints lead or mercury or tin? And the reason these heavy metals were there was because what happens is that if you have mold or bacteria starting to form on the surfaces, you would have them inhibited from growth because of the presence of those heavy metals. Why do we embalm bodies? Embalming the bodies with urea, formaldehyde, you can do it also with methanol, basically stops the enzymes, the proteins, that would putrefy the body, that break it down. You want a real simple example of all of this? Egg white. Raw egg. Notice that it's transparent. Notice that it's liquidy. Okay. 2 properties. Cook it, apply heat. What happens? It becomes opaque. It becomes more solid. So what you've done is you've denatured the proteins that make up the egg white. And as a result, they've lost their transparency property. They've lost their flow property, being somewhat more on the liquidy side. Okay? So we've gotten through the first lecture. Great. Well, here's a couple of things that I do always. I try to tell you how to review and prepare for this. We will be having, by midweek, Mastering Microbiology all set up and running. The chapter review at the end of the chapter, review pages 1 to 16 and pages 51 to 59. Review the visual summary on pages 29 and 61, relevant content only. Review the chapter overview on pages 30 and 63. Review homework for Mastering Microbiology, the quiz for Mastering Microbiology, and peruse videos in the Blackboard for the lecture content. There's a lecture file there. I will have always a lecture and a lab file that'll have a bunch of related videos that will help expand your understanding of the topic. The next section we will be going through is Introduction to Prokaryotic Cells, which is in Chapter 3. Have a nice day.